welcome everyone. I have the pleasure of introducing Paul Borghesani, who is one of my colleagues. He's an MD, PhD. He's assistant professor in the Department of Psychiatry and Behavioral Sciences at the University of Washington Medical Center. He obtained his PhD in basic neurobiology, studying how neurons are born and mature in the developing brain. As a psychiatry resident at UW, he turned his attention to using neuroimaging in the study of aging and cognitive impairment. The overall goal of his research is to understand what constitutes healthy or optimal cognitive aging and how we can use neuroimaging to differentiate between benign age-related cognitive changes and early dementia. He began working with Dr. Shea and Willis and the Seattle Longitudinal study, which you'll be hearing about today, in 2008 because it was a natural fit to pursue these questions. Dr. Borghesani.
Thank you. And I want to thank Dr. Ram and other folks from the department for asking me to talk. It's always nice to talk to the home crowd. I don't think I've ever really presented in a grand rounds fashion here my research. I've done many other presentations that are more overviews and reviews of the field. Today is not an overview or review of the field. It is really primarily just my results and what work I am doing. So, well, present is about 1/3 old, about 1/3 recent. And when I say recent, I mean papers are either nearly in press or have been worked on in the last year or so, and about 1/3 current. And what I mean by current, I mean is that I'm still Skyping with people now talking about what the results are. And you'll hear about many different types of neuroimaging here. I did some structural imaging, basically looking at the volume of certain regions in the brain. I've done diffusion tensor imaging, looking at white matter structure. In addition, we've done some functional imaging most recently to look at how the brain is connected in a more functional way. What I want to do when I present all this material is have you understand some of the methods enough so that you can appreciate what the findings mean and why we are doing these things. So there's a little bit of technical stuff, but not very much. And I will be presenting some cross-sectional data in addition to longitudinal data, so don't be surprised by that. So there's lots of slides I could show as sort of a general overview about aging and cognition. And I choose this one because I think it's the simplest and, to me, the most meaningful. It just comes from the Alzheimer's Association right off their website, and it just shows the change in the number of deaths between 2000 and 2008 in some major diseases. We have breast cancer, prostate cancer, stroke, and Alzheimer's disease. Now you see the giant purple bar that's going up in percent. That's Alzheimer's disease. And many folks will say, oh, well, their population is aging, so there's just more Alzheimer's disease. Well, that's very true. But the aging is also the primary risk factor for breast cancer, prostate cancer, heart disease, and stroke. And yet they're going down, the number of deaths. So clearly, while we're making advances in treating other diseases associated with aging, we're not doing super well in learning how to treat the degeneration of the brain. So when we talk about cognition, and today I'll be talking exclusively about normal aging and not dementia, but in order to understand dementia, we need to understand the normal cognitive processes. I always start with this sort of canonical slide, and anyone who's heard me talk has seen this one before. And what it is looking at cognition from two perspectives. And the abilities here listed are not important. But as you see in this leftmost graph, the abilities seem to go down from about age 25 straight through to 81. Some of them are a little flatter. Their numeric ability and verbal ability are a little flatter. In the right graph, what we can see is that the abilities seem to be a little bit more stable throughout what we'd say midlife, age 40 through age 65, 70 or so. So why is that? Where does this data come from? And where it comes from is that The data on the left is cross-sectional data, and the data on the right is longitudinal data. And this is well known for cognitive studies, and something that has been explored for many decades. There are lots of reasons for why there might be these cohort effects or differences between cross-sectional and longitudinal studies. But what I want to focus on is just the idea that it's important for our understanding of cognition and aging to know actually where people have been, because simple cross-sectional data doesn't provide that. And it's clear from our longitudinal cognitive studies that this information is important. So the study I'm involved in is in very much collaboration with Werner Schei and Sherry Willis, who developed the Seattle longitudinal study in 1956 and are still running it. This study was developed by recruiting 500 people in 1956 of mixed ages. So what we see here is where the recruitment date and the age of individuals, and here's the birth dates of those individuals. Every 7 years, Dr. Schei and later on Dr. Willis returned here to Seattle to recruit an additional 500 participants or more, and then follow up with the 500 previous. So over the years, this has been done every 7 years, such that at this point, we have lots of longitudinal cognitive data on individuals. In 2007, a little earlier, like 2005 actually, they started a neuroimaging study. And what they did is they selected individuals of mixed ages. So individuals born from between 1914 and basically 1955, and actually a little later now. So those individuals were all imaged at the same time, but they were different ages, and they had different amounts of longitudinal data in the set. The longitudinal imaging study that I'm going to be talking about was focused on what happens here during midlife. And subjects were specifically recruited who had substantial data from midlife on their cognitive trajectories. Who are these subjects? At this point, there's 188 plus subjects that have had neuroimaging. They come from a larger pool of over 1,200 participants in the Seattle longitudinal study. Their average age is about 70. They range from 54 to 89. A little excess of females, but more importantly, it's a very well-educated sample. The majority have a college degree. A vast majority are Caucasian. And surprisingly, very high carriage of the ApoE4 allele, which is a risk factor for Alzheimer's disease and many bad things that happen in the brain. They are cognitively intact. These are not demented individuals again. Demented individuals generally drop out of the study, and we do not follow up with them in either the cognitive exams nor the neuroimaging. If we look at this group, it's generally fairly healthy. They have very few vascular risk factors, meaning diabetes, hypertension, hyperlipidemia, smoking. They also have very low levels of white matter hyperintensities, which are a marker in the brain of sort of vascular pathology. And if we score these by hand or if we do more automated methods, we see that they generally have healthy-looking brains. So just a couple examples of some of the cognitive data and findings that Dr. Schei and Willis have generated over the years that I think are important to recall when we're studying sort of longitudinal aging samples. The first is that the trajectory of cognitive change in an individual is very dependent on factors in that individual's life. For instance, here, education and occupation. It's hard to read this, but if we look at only a grade school education, you see this sort of trajectory longitudinally as people age, and this is for verbal fluency. Verbal memory. Whereas the higher education you have with some college or high school, some college, or graduate school, the trajectories seem a little different. It's not just that they are people are said to be higher functioning, it's also that the stability seems to last slightly longer. This is true for occupation too, in that unskilled sort of workers, which are very hard to recruit these days, seem to have a quicker rate of decline during sort of the midlife period than more sort of white-collar occupations. Another finding that I think is even more interesting is there seems to be a change in the general population's ability to perform these cognitive tests that's based on when you were born. Now, this isn't really that surprising. It's just interesting to document. So this is essentially looking at people born in 1903 versus people born in 1973, and it's normalized to essentially a standard age, and all the cognitive tests are the same. And what it's showing is that, well, people in general are getting better at a lot of these tests, excluding a few things like this is numeric ability and verbal ability, which seem to be slightly on the decline. But what just shows is there's some instability in how well people do in these tests based on when they were born. This is, I think, most dramatically illustrated in this nice slide, looking at parents and their adult offspring. And this is parents at age 60 and 74 and their adult offspring at age 60 and 74. And what we see is that it's very, very different, with generally better abilities and also just less decline. So this type of sort of difference between cross-sectional and longitudinal findings isn't just in cognitive data, but it's also in neuroimaging data. And one of the more surprising recent reports out of the very large Alzheimer's disease neuroimaging initiative that was reported this summer was that if you looked cross-sectionally at the thickness of an area of the cortex called the entorhinal cortex, this is an area in the medial temporal lobes that's very associated with Alzheimer's and Alzheimer's disease. If you look at how this sort of atrophies or gets thinner over age in a cross-sectional way, you get sort of these lines as you do the regression analysis. However, you follow up these same subjects and scan them a second time and ask, how does the thickness of the entorhinal cortex change? You get dramatically different estimations for how quickly atrophy is going on. Now, this is actually very different than the cognitive data. The cognitive data would imply that there's less change longitudinally than there is cross-sectionally, whereas here we have more change longitudinally than cross-sectionally. And this has yet to be completely explained, but one of the things that certainly goes into this is that older subjects, recruiting people to neuroimaging studies means you have to be fairly healthy. And so essentially the older subjects here are really the healthiest subjects and very much might have been maintaining their brain size. So the overall goals for the talk today is to sort of show how current cognitive abilities and past cognitive change are very really related to brain structure and connectivity, and also to explore how functional connectivity is evaluated by MRI is related to cognitive abilities. So let's start with some of the old data, and we're talking about hippocampal volume and midlife cognitive change. So the hippocampus is part of the medial temporal lobe. It lies just below the entorhinal cortex, and it's the area that's very associated with Alzheimer's disease and other disorders of memory. We asked two questions. One question is hippocampal volume related to past cognitive change, specifically during midlife, and also how specific is this association? Meaning, is it related to certain aspects of cognition or all cognitive variables? In the background here, in sort of the faded, I just showed the hippocampus. This is a painstakingly dissected hippocampus, which must have taken hours. And this is, of course, a seahorse, and that's where it gets its name. I think it's a great image of the hippocampus. So here's our canonical slide of longitudinal cognitive change, and we're focusing on this midlife region. And I told you it's fairly stable in longitudinal studies, specifically the Seattle longitudinal study. In truth, though, if we look at individuals, there is some variability. Some individuals improve. Some individuals actually decline, and some individuals, or most individuals, remain relatively stable. For this neuroimaging study, Dr. Willis and Schule selected individuals who were changing in 3 different midlife abilities. 1 being memory, 2 being executive function, and 3 being flexibility. And let me just talk a little bit about the cognitive measures using the SLS. There are many of them, so I'll just give you a selection of them so you know what I'm talking about when I talk about memory here. For memory, there are 2 measures, immediate and delayed recall, very standard tests of 20 words that have been memorized after 5 minutes for immediate recall or greater than 30 minutes for delayed recall. For word fluency, again, standard, just generating S words in a period of 5 minutes. For reasoning, it's detecting patterns in letter sequences, and this is kind of an odd test, so I'll just show an example of this. Literally, you're shown a string of digits, letters, and asked to fill in the blank. And as you can see, if you go across A, B, X, C, D, X, E, F, X, G, H, X, what's the next one is I. So you'd have to figure that out, and there's multiple sort of examples of that, and this is how they get the reasoning score. For cognitive flexibility, you're presented lists of words and asked to generate either antonyms or synonyms, but you're asked to generate them based on the capitalization of the word. So what you have to do is sort of think, you see the word, decide if it's in capital or small letters, and then generate an antonym or synonym based on whether it's capitalized or not. So this shows your ability to sort of do a lot of shiftings, and we also call this set shifting of kind of cognitive processes. Finally, perceptual speed has been measured, and that is sort of very basic comparing pictures and matching, finding capital A's in a paragraph, and comparing numbers. So the subjects, 165 in this case that were selected on these change on these measures, we see that about 15% for all abilities either improve or decline, and the majority, around 70%, remain stable. Now these are the same people, so some people can improve in memory and decline in executive function. The subjects for the studies I said were ranged in age from age 56 through the late 80s. So they either selected immediately after that midlife period, essentially right after the change occurred, or they could have been selected 20 years post that change occurring. So what we found in our initial study is just looking at hippocampal volume is that improvement on memory was associated with stability in hippocampal volume. So looking at hippocampal volume of younger individuals who had changed memory scores, so this is decline, improve, or stable during midlife, we see that they all have very similar hippocampal volumes. There's no difference. If we look at older individuals who have either declined, improved, or were stable on memory function, we see that the ones who improved during midlife actually have some sort of stability or less hippocampal atrophy. This is just illustrated here for all the subjects in the study. The solid line here going across is those individuals who improved in midlife. Now, just to give a perspective for how this fits into the overall literature on hippocampal volume, these are cubic millimeters change. So the difference here is about roughly 1,000 cubic millimeters. If we look at a recent meta-analysis of all diseases in hippocampal volume, it's very interesting. I just pulled this last week. Diseases like hypertension, obesity, and diabetes actually change or potentially change hippocampal volume very minimally. Curiously, there's a lot more heterogeneity in our psychiatric diseases. This is the actual data. You've probably all heard that depression causes hippocampal atrophy. And this is actually the number of studies that have done that and looked at that and what the findings look like. It might be less impressive than what you read in the New York Times. Same for adult onset PTSD or childhood onset PTSD. If we look at just the number of 1,000 millimeters cubed, or we see that it would fall very much in the high range of all these studies. So at least from the perspective of comparing it to other studies, it looks like midlife memory improvement is more important for sort of the volume change of the hippocampus than many other things. So summary from this is that midlife memory improvement is related to stability in hippocampal volume. And I should say that other abilities that we looked at, including current abilities, were not related to hippocampal volume. So let's change gears a little bit and look at white matter integrity and cognitive aging. We're going to ask the same sorts of questions. How is white matter integrity related to current cognitive abilities? And is white matter integrity related to past cognitive change? Again, the same sorts of questions. So let's just pause and talk about white matter. White matter really doesn't get much respect. It has not been studied very much. And there's a very good reason it hasn't been studied. It's difficult to study. You can't trace a neuron, you can't trace an axon without very specialized methods. And even if you do the specialized methods, you have to cut the brain very, very carefully in order to trace axons. It's a very difficult job to do. Secondly, when you cut the brain, it's hard to assign sort of, well, what part of the white matter is this? I mean, you could get a general impression that it's in the prefrontal cortex or something, but it's hard to define it clearly within a subject or even across subjects. So, in truth, white matter has been largely ignored. I mean, everyone knows it's important. Connections are important for the brain, of course. But it's been much less studied than cortical thickness or atrophy. Now, if we look at a very sort of old sketch, and I wish I looked on the web to find out who actually drew this. I think it's from like the 19th century. These are some of the major white matter tracks, and I'll actually be talking about some of these, so I just wanted to give you an illustration. So connecting areas of like the prefrontal cortex, the parietal cortex, and the occipital lobe is the superior longitudinal fasciculus. And you can't read the name, but it's right here. Connecting sort of the medial regions of the prefrontal cortex and the medial regions of the temporal lobe is the cingulum, or the cingulate gyre. The cingulum runs right down here over the corpus callosum. Connecting the occipital lobe to the temporal lobe is the inferior longitudinal fasciculus. And finally here, connecting sort of the ventral prefrontal regions and the anterior temporal lobes is the uncinate, or the uncinate fasciculus. So how does DTI, or diffusion tensor imaging, look at white matter integrity? Well, it's kind of a neat trick. If you look at axons within white matter, they're often oriented in a similar fashion, kind of like logs lying in the white matter. And diffusion of water is different along the axon. Now this picture shows within, and I don't mean to imply it's only within, but along kind of the perpendicular way of the axon, and it is across the axons. And so a group of axons within the white matter will have a different amount of diffusion depending if you're talking the long ways, axial or radial. And from diffusion tensor imaging allows you to map this diffusion and generate a lot of metrics to describe this sort of ellipsoid, cigar-shaped structure. Now, this cigar-shaped structure can take various shapes. It can be much more spherical, or it can be more like a football, or it can then be more like a piece of rice. And in order to describe this, several measures are commonly used. There are alternatives to this, but these are the commonly used ones. The first is axial diffusivity, or AD, and that's along the major axis of this ellipsoid. The second is radial diffusivity, RD, and that's along the minor axis. The second two are the mean diffusivity, and you can, as you guessed, the mean is just the average of all the diffusivity within this ellipsoid. And the final one, which is probably the most studied and most used, is the fractional anisotropy. And fractional anisotropy is a ratio, and it's not a pure ratio, it's a mathematical formula, but it's essentially can be thought of as a ratio of the amount of axial to radial diffusivity. So it gives you an idea of what the cigar shape is. How cigar is it? A perfectly long, skinny cigar would have an anisotropy or fractional anisotropy of 1. A perfect sphere would have a fractional anisotropy of 0, because diffusion is equal in all directions. So by calculating this within voxels of the white matter, you can actually construct the direction of axons. So here is a typical image. This is a fractional anisotropy image. And you can actually blow up a region of this and look at the voxels and assign how big are these, how much diffusivity there is and what direction it is. And from this you can sort of map axons through the brain without actually doing those cumbersome anatomical procedures. Now, it's not perfect in the sense that it's a mathematical recreation. So these vectors don't prove connections. It simply suggests the overall pattern of white matter of fibers. So if we look in our sample of about 188 in this image, individuals, on the changes associated with aging in the white matter, we see this. And this slide is meant to be a Gestalt slide. Clearly, there's a lot of, it's very hard to see. But what it shows is regions where the fractional anisotropy, or the cigar shape, is being reduced in aging, and regions then where the MD, the mean diffusivity, the axial, and the radial diffusivity are increasing with aging. And this is a typical aging pattern reported in dozens of papers. What does this mean? So why does this happen during aging? Well, generally thought is that the white matter structure is breaking down. The organization of white matter is breaking down, such that the cigar shape, which represents a lot of organized structure, is being reduced and becoming more spherical. In addition, there's loss of axons, probably loss of myelination, an increase in the size of the extracellular space. That would all just increase the amount of diffusion in sort of all directions, and thus the increase in mean axial and radial diffusivity. The other thing to notice is that the changes are throughout the brain. It's not localized primarily to only one region. And although some have characterized sort of a anterior to posterior gradient or a ventral to dorsal gradient, overall there's lots of white matter change throughout the brain. Now, if we look at our cognitive variables and say how are they related to white matter integrity, after accounting for age, of course, it's much more restricted for some of them. For instance, reasoning, which is primarily thought to be an executive task dependent on the function of the prefrontal cortex, is specifically associated with, in this case, it is axial diffusivity within the left prefrontal cortex and not anywhere else. This is good. This is where we think it should be. It's nothing very astonishing, but it is true that many studies haven't found this before, probably because the size that we're using, 188 subjects, is much larger than most studies of aging. However, if we look at other cognitive abilities, for instance, our perceptual speed or our cognitive flexibility, and I apologize, every time I say flexibility, I mean cognitive flexibility, so if you do yoga and you're thinking the other, no. So if we look at processing speed, perceptual speed or flexibility, we see that first they're related to white matter integrity in a much larger proportion of the brain than reasoning was. That's the first sort of take-home message from this slide. The second take-home message is that they're sometimes differently related, in that perceptual speed appears to be related to this sort of white matter, this fractional anisotropy, which is not related at all to our flexibility. Whereas the mean, the axial, and the radial diffusivity are sort of related to all, to both parameters. So again, this wasn't too surprising to see, and although many reports didn't say exactly the same thing as we found, it is what we would have generally expected. So we went on in the study to ask another question, which is, does white matter integrity account for the association between different cognitive parameters during aging? So if we look at our canonical graphs here, and we see this green line, which is perceptual speed. As you notice, the green line, perceptual speed, actually goes down pretty much throughout life in both longitudinal and cross-sectional data. And perceptual speed has been hypothesized by some to be primarily responsible for all cognitive change during aging. In other words, it's not really that people are unable to reason or aren't really flexible or aren't able to, their memories even perhaps are equivalent. But all of these other cognitive abilities are dependent on speed of processing, just encoding words, especially on the tests that neuropsychologists administer. So there's some who say that perceptual speed accounts for most of this. Within our data, we actually see some curious things between perceptual speed and some of our cognitive measures. For instance, if we just look at our reasoning abilities and where it's associated with white matter integrity, and perceptual speed where it's associated with white matter integrity, we get very different patterns. Perceptual speed seems to be associated with very large white matter tracts. This is the internal capsule, millions of fibers, and the corpus callosum. Whereas reasoning seems to be more associated with more distal areas of the white matter. Now, I'm not sure where to go with this. And I actually removed it from the last paper that we're hopefully getting accepted soon. But it's a curious thing, and no one's reported this. But to get more at how perceptual speed is related, we want to do what's typical in the aging field, some mediation analysis, basically asking how much of the variance can be attributed to white matter integrity versus other cognitive variables. And what we did to do this is we have to extract average values. So we looked at regions of interest within the white matter, and here are those names that I showed you before, just so to introduce the inferior longitudinal fasciculus, superior longitudinal fasciculus cingulate, the uncinate, and then the anterior thalamic radiation, which is here, basically is containing white matter or axons connecting the thalamus to the prefrontal cortex, and also a lot of descending fibers. So when we do that, what we did is we extracted average values for these three, I just had to remind myself, 13 regions, because these are all bilateral. And we had 4 DTI parameters, so 13 regions and 4 DTI parameters. We had 52 different values within our white matter metric. And in a very kind of non-hypothesis-driven approach, you throw them all into the same model. And that's kind of fun statistics. You put in 52 variables, and you ask your program to do stepwise regression, throwing out the insignificant ones, and to generate a model that explains the most variance in whatever your dependent variable is, in which case we were trying to explain our cognitive abilities. So when we did this, many regions came out, which is not surprising. And we were able to explain variance in our cognitive abilities. What was kind of surprising is that the amount of variance explained by white matter integrity was equal or greater to the amount of variance explained by age. Oop, looking at the wrong thing here. So here's the amount of variance in reasoning, close to 20%, explained by white matter integrity compared to age, just looking at the heights of the bars, or for flexibility. again, others had done this sort of thing and found that to control for age, white matter integrity didn't really contribute anything new. So this kind of went beyond what others were reporting in the field. But in addition, we asked the question, well, how much of the association between processing or perceptual speed in our cognitive abilities can white matter integrity account for? And likewise, how much of the age association in our cognitive abilities can white matter integrity account for? So then we look at the colors here. And what we see is for age, about half of the variability that we would attribute to general aging could be accounted for by white matter integrity. Not a lot, I mean, just about half. And this is true for both reasoning and flexibility. For perceptual speed, the results were very different, in that most of the variance that could be accounted for by perceptual speed in reasoning really couldn't be attributed to changes in white matter integrity. So there's something different going on here. We couldn't simply say the perceptual speed, which was thought to be dependent on white matter integrity, was simply kind of working through that mechanism to affect reasoning. In contrast, the amount of variance that is explained by white matter integrity is regarding the amount of the amount of the variance, the proportion of variance explained by processing speed. About 50% of it could be attributed to white matter integrity when you're looking at flexibility. And so what this shows is that the relationship between some of these factors, such as processing speed and white matter integrity, differs for each cognitive domain. So just in kind of a summary of this was that age is associated with loss of white matter integrity, and this is well known. Reasoning is associated with white matter integrity specifically in the left prefrontal lobe. So there's highly selective regions of the brain that are associated with certain cognitive abilities. Other cognitive abilities are much more generally associated. And what I didn't show you is that many of the cognitive abilities are not associated with white matter integrity at all, such as memory or word fluency. And secondly, the amount of variance in other cognitive abilities explained by processing perceptual speed is variably accounted for by white matter integrity, meaning that there's something much more to it than simply saying, well, perceptual speed is decreased because axons are dying, and that's why it counts for all the other cognitive changes. So in some other work, we asked the second question, which is, well, how does past cognitive change relate to white matter structure? And to do that, we had to use a much more complicated model in which we put several variables in, including the cognitive variables, age, the interaction terms, and then midlife change in those parameters. And also the interaction between midlife change and cognitive, all the interactions essentially. And then controlling for gender. And what we found is that, generally speaking, there wasn't an association, which actually wasn't too surprising. We were looking for something pretty subtle here. However, for flexibility, we got something that very much surprised us, which is that there was a large association with change in flexibility during midlife and white matter integrity if you accounted for their current flexibility, their current ability on these flexibility tests. Now, what was surprising is not that we found this, because lots of little blips and distals kind of show up when you do neuroimaging analysis. What was surprising was that it was massive. It was essentially the entire brain. And when we looked at it down here, we found that like 42% of all of our voxels had uncorrected p-values of less than 0.1015. If you are a statistician and you think of that, well, that many of your voxels have that p-value or less than that p-value, it's massively statistic, and likewise for the mean diffusivity. So what we did, or what I did, is something that was so basic, is I just took one single value. I just averaged across the entire brain the fractional anisotropy and the mean diffusivity. So I get a single number or two numbers per individual and ask how is that related to their midlife cognitive change, their current abilities, the same model. And that was also very significant. So this is the interaction term between score and midlife change. Now, interaction terms are very difficult to understand, so that's what I want to show you here. Yep.
So skeletal means the white matter skeleton?
The white matter skeleton, which contains about 100,000 voxels. So of 100,000 individual tests that were done in this prior slide, 42,000 were significant.
And the voxels in the 42% mean, what are you saying about that 42%?
We're getting to that. So what do we mean by that 42%? Okay. So in order to understand what these interaction terms mean, you really have to do some modeling, because you have to put some numbers in. And this is what we see. So looking at a calculated value, so this is not real data. This is taking the beta values generated in the prior model. If you're not doing well on your flexibility scores, it turns out that it doesn't really matter if you improved or declined on flexibility during midlife. Your fractional anisotropy goes down with age, as it typically does, and your mean diffusivity goes up with age, as it typically does, and there's nothing statistically different between these two lines. However, the model would suggest that if you are currently doing very well on flexibility, that it's actually very important how you got there, or what your past history was. And here, what we see is both these graphs, one for fractional anisotropy and one for mean diffusivity, look at people who are functioning well on flexibility. However, the solid line are those people who actually improved in flexibility during midlife, and the dotted line are those people who declined. So what's happening here? What's happening is that, or I can't say I know what's happening, but I can say that what our thought might be is there's some sort of compensation. So individuals who declined in flexibility during their midlife are able to maintain, some of them at least, are able to maintain a relatively high performance level only because there's some sort of compensation for the pathology that's occurred. So this is sort of in support of this idea that some people, even if they have significant amounts of brain pathology, are able to compensate for that by using some sort of alternative systems. So white matter integrity and past cognition is the past change in flexibility is related to white matter integrity during aging. And it appears to be dependent on current abilities. And it's specific to that ability and widespread throughout the brain. Finally, I want to tell you about some of our most current stuff, and that is looking at functional connectivity. And we ask the same sorts of questions. Is functional connectivity associated with past cognitive change? And we're very interested in that, and this study was designed to do that. And then we also looked at different connectivity measures, because that's what I'm interested in. So functional connectivity, and I use this slide because it's actually truer than life. We ask people to get into the MRI, get hooked up to all sorts of monitors, and literally just to relax and think about nothing. They, of course, are not relaxing, and they're not thinking about nothing. They're thinking about all sorts of different things, and we don't even know. And last time I checked, the French developed a questionnaire of 250 questions to try to understand what people were thinking about in the scanner. I have not administered that test. So what is the functional MRI method? So there's two very basic ways of doing functional MRI. The one you read about in the New York Times several times a week, it seems, or no longer that much, but is that you use a task. So essentially, if you look at time across the x-axis here, you do something. In this case, the task is just opening and closing your eyes. And then you record across time from the brain, and certain areas of the brain will light up or show changes in blood flow / blood oxygenation. In this case, of course, it's the occipital kind of primary visual areas that are activated by this simple task of opening and closing your eyes. The alternative way of doing resting state or functional MRI is to do what's called resting state, which is basically have them in the scanner and doing nothing, just lying there. And you collect a time series. And then from that time series, you simply ask, how are the voxels in the brain correlated with each other? And I've talked about this before in this forum. But it's fairly revolutionary when people first thought about this, because no one expected it to work. If you do this sort of analysis, you identify specific networks. And this has been replicated many times at this point. Some of them are very much expected from what we know, sort of the primary motor, visual areas, these attentional areas that are in the prefrontal and dorsal parietal regions. But some of them are sort of unexpected, and this is called the default mode network, which involved medial regions of both the prefrontal and parietal cortices and also dorsal lateral parietal cortices. And if you look at these networks that are identified by this method, what you find is that they can, they don't always, but they can sort of interact in sort of reciprocal nature, with one network being online when the other one is off, and vice versa. So this is just a picture of one of these networks, the default mode network, in red, and this is sort of a motor network in blue. And what's shown below is the time series of the red and yellow network compared to the blue network. And it's showing that, at least across time, that these networks can be characterized as sort of fluctuating in sync with each other, sort of anti-correlated. Now, I'd point out on this that the time here is long. This is 250 seconds if you look at this. So this fluctuation, 250 to 300, let's just say it goes to 300, is 50 seconds long. So these systems are not exactly fluctuating quickly. If it's every 50 seconds, it can't be involved in real complicated thought, because thought is much, much faster than that rate. So with this sort of background, what I did a couple years ago is did a small resting state MRI pilot project, and we recruited additional people from this SLS. And we recruited them specifically in midlife, so younger individuals, and who had changed in executive function, either declined or were stable / improving. And I will say that slash here, we combined the stable and the improving because we weren't convinced we had improvers. But we recruited 29 people and did some functional imaging. And these are the people, their average age was 60, so a little bit younger than the other group. Their ApoE is very similar. These are brain volumes are no different. Again, white matter hyperintensities are no different. Their abilities are different. As you imagine, we got them at the end of midlife. They're younger. So folks with declining executive function had lower reasoning, lower cognitive flexibility, and word fluency. And if you did some sort of composite executive function, you can see that was lower. And there was a difference in change. So we asked, are these resting state networks affected by this cognitive change? And before I get to it, let me just show you two more pictures of resting state networks to show that first, they are very much reflective of cognition. So these are the 29 subjects that I just showed you doing the cognitive task. And a cognitive task here is a very simple one, just deciding if a dot is above or below a bar and how far away it is. It's very simple. But the areas that are activated during that task, shown in red, versus the areas that are deactivated or get less activity, shown in blue, are on top. And below is a resting state scan from the same subjects, different data here, same subjects, different data, just looking at correlations. And as you can see, the resting state networks mirror the networks that are identified in a cognitive task very closely. So there are various ways to identify resting state networks. The two most common are sort of seed-based or ICA, but I won't get into that at all. But what essentially you do is just look for correlations. So you can either take a single voxel. Excuse me. You can either take a sort of an average of multiple voxels and ask how are they related to individual voxels throughout the brain, or you can take a single voxel, and I put a big X through it because it doesn't work very well, and ask how a single voxel relates. And the reason why it doesn't work very well is if you take a voxel right next to the one that uses a seed, you can get very different results. And so people don't generally like this method. The other way is just to take the average of multiple voxels from two different regions and ask what the correlation is. And that's fairly simple and more straightforward in many ways. So we did this in our subjects. And what we did it for is the network, the default mode network, which is sort of the canonical resting state network, and also very involved in aging and dementia. And we took our canonical default mode network and we segmented it into nodes and basically just created a model of 6 nodes and 15 connections. And we asked a simple question, okay, how is that network connected up? And how does it differ between the two groups? So if you look at using a seed region, which is basically just taking the average activity within the medial temporal lobe, using it as a regressor against all the activity in the brain, ask what's connected to it, what correlates. What you would conclude is that people who decline in executive function actually had greater connectivity between the left medial temporal lobe and medial prefrontal cortex. And the image of that is just here. And if you extract values just for explanatory purposes, you see this, that the people who are stable are down here. So here's their current executive function. These are individuals who are stable. These are individuals who decline. They seem to have greater connectivity. Now there's something about this graph that very much upset me when I saw it. And that is that greater connectivity was associated both with decline and with better executive function. So you're saying that connectivity, or higher connectivity, was with a marker of better function and decline. And that's a little confusing, and it didn't make any sense to me when I got the results. We then used a different method, which was just the ROI method, extracting average values from multiple regions. And what we found is that there was a single connection within this model that was different in our subjects. And it said the same thing, is that the decliners actually had greater connectivity than the ones who were stable. And again, graphed in that graph should look somewhat similar to the last one. So it ends up with a model that said this, that decliners have greater functional connectivity in select connections of the default mode network. And it's only these connections, it's not other connections. So it's connections for the left medial temporal lobe to the prefrontal and posterior cingulate cortex, and then connections between the two. But also for those connections that better executive function is associated with better connectivity. So again, I was unhappy. Now this is sort of standard ways of doing connectivity analysis. So I want to come back to this, the method that I said was not good, using a single voxel. So what happens if you use a single voxel, as I said, if you move one voxel over, you get very different results. And people argued about this for a long time. Well, then how do you use a single voxel? Should you just average them? However, I kind of thought that, well, that must be important. If shifting your location by 1 millimeter in the brain changes what your connectivity is, somehow that seemed important to me. So I approached some engineers, actually, within the Integrated Brain Imaging Center here at the University of Washington, and asked them, could we start looking at voxel-wise connectivity measures? Rather than taking these average regions, or could we take a single voxel and say, how is that single voxel connected to single voxels in another region, sort of intER-nodal? Or how is that single voxel related to other voxels within the node, intranodal? And if you do that, you can define a few terms. So intranodal connectivity, which is the average of all the voxel-wise correlations in the node. You take the top 10%. Internodal connectivity, the average of all the voxel-wise correlations between two nodes, and the top 10%. And you get a very different story. In fact, what you get is that the intranodal connectivity, or the connectivity, is actually less within your medial temporal lobes, both left and right in this case, where the right is like the p-values of 0.06. You take the highest 10%, same finding. And likewise, if you look at the internodal, so the between-nodal connectivity measures, what you get is a very interesting pattern, and actually replicates, but in the inverse direction, of our other findings, which is if you look at the left medial temporal lobe, it has less connectivity with the medial prefrontal, the posterior cingulate, within itself. So this is the intra, and with the right medial temporal lobe. Likewise, if we look down these columns, what we see is it's the left and the right medial temporal lobe that seem to have all the significant p-values, or near significant. So if you put that together, what you get is this sort of model, which is within our decliners, we actually have less functional connectivity within the left medial temporal lobe and between the projections of that with the posterior cingulate, the medial prefrontal, and the right medial temporal lobe. Now, when you actually then look at these values in the same way we did before, what you get is something that actually makes some sense, which is that people who are stable, there isn't really much association between the connectivity and their current executive function, but connectivity is lower if you've declined, and maybe related, but I wouldn't certainly trust that R value at all. And this actually makes some sense and isn't counterintuitive. And in fact, if you look at the two models together, they're actually inverse. Essentially, they both identify the same regions as being involved in midlife decline, but one would say that they are greater connectivity and one would say they are less. And this got me thinking about this slide as far as sort of the antagonistic or anti-correlations that are seen in the big networks, that perhaps we were looking at networks that were also somewhat reciprocally. So if you look at connectivity from a voxel-wise level versus a regional level, you might actually come to very different conclusions, and they might be it might be important to see how they're related to each other. So finally, midlife executive change is associated with both intra and intER-nodal connectivity of the left and near right medial temporal lobe, and that is certainly consistent with what's known in the field. And differing resting state MRI methods can lead to very different interpretations of your data. I want to end with some overall summary is that specific cognitive abilities are associated with select regions of brain aging, and I think this is important. That's hopefully what I demonstrated with some of the initial work. Such that if you're having trouble with a certain area of cognition, it's very possible that there are differing regions in the brain that are affected versus another person who's having a different issue with cognition. And secondly, the midlife cognitive change may be predictive of brain aging trajectories. So it's not just how you're doing when you're age 70 that's important, it's how you got there. And this is certainly very well known in that clinically, of course, if we have a mini mental status exam of 26 in a neurosurgeon, we would worry about them. And perhaps that wouldn't be so worrisome in other folks. And finally, functional connectivity may be sensitive they may be a very sensitive method. I mean, this is 29 subjects. We're now currently repeating this with our larger sample of 188 individuals to see if using this novel way of looking at brain connectivity on a voxel-wise approach it actually holds up. So I just want to identify people that I work with, primarily Sherry Willis and Werner Schei, and the rest of the people at the Longitudinal study. And recently, with Art Chavalet-Wangzi and Gianluca Gazzola, who are doing MATLAB programming for me so that I don't need to do that, to determine the intra and intER-nodal connectivity. So thank you. And I think we have I don't know if we're actually over, but I think I've used my time for questions if there were any. I'm sorry. I'm here if you have questions.